Today I'm going to give you a kind of a preliminary results from our Pierce's disease biocontrol technologies that we have been trying to use to control in California. This is the field trial that we have done it on our campus at UC Davis. So before I start my talk, I would like to acknowledge our collaborator. First of all, Molly Araquin, who is a technician in my laboratory, who was responsible for all these trial, and then she has done an awesome job. Also would like to thank to Anika King Hapwala, who is the representative of the sorry about that. The treatment that we have applied, which I'm going to talk about it, Philip Rosausen and Steve Lindo are also collaborative on this study that I'm going to talk more about what their involvement were. I just want to give you a little bit background information. You all know that the Pierce's disease caused by the Xylella fastidiosa, now we are calling it subspecies of fastidiosa. It is a gram-negative, slow-growing bacterium which is colonized plant tissue, specifically xylem, which is that they also call it the xylem-limited bacterium. Because of that, the symptoms are look like the drought. That I'm going to show you some of those information. Anyway, the disease is called Pierce's disease. That's name came from the scientist who has first started working on this bacterium a long time ago. So Pierce's disease has a long history in California. It has been first found in 1880. For last more than 100 years, it has been causing tremendous of damage. Especially in Southern California, where we have the effective vector, it has changed the agriculture of the grapevine industry, and then here we are still dealing with this disease. So here's a couple of the pictures to give you an idea how these bacterium could colonize in the xylem tissues. These are some of the publications that I found. So the one on the one to see that you are here is that you see a lot of bacteria. This is the xylem vessels. When they plug in the vessels, they just plug vessels, as you can see physically. So the picture on the right is also showing a good colonization of the xylem vessels. So there are a lot of studies that has been done by my colleagues in UC Riverside, who has been demonstrated that these bacterium could degrade the pore membrane in the xylem vessels. They can move physically up and down within the plant tissue, which makes it this disease progress within a short time within the plant. So I'm sure you all have heard about it. Xylella fastidiosa has a wide host range. So far, more than 303 species have been identified as a host in California. Vitis vinifera, which is grapevine, is one of the major problem. It has been also found on citrus. It hasn't been confirmed in California, but in other parts of the world it has been found, which in California it's a big concern. We have a lot of citrus orchard. Besides the commercial crops, it has also been found on the landscape and other native plants. Liquid amber is one of the most common host species that I myself have been coming across and then seeing a lot. So in terms of the inoculum source or distribution and host, this bacterium doesn't have any problem for moving from host to host and then surviving in California nature and agricultural areas. So here's one of the distribution map that I was able to find of the disease in California. As you can see, all the blue lines has been confirmed This is not the latest. The latest date was that the 2007, which I'm sure it has been progressed a lot. So just to give you an idea how much this disease progressed in California. So this disease is vectored by the sap-sucking insects. Glassy-winged sharpshooter is one of the major the vector in Southern California, which is one of the most effective vector. This is again one of the old the map that I was able to find in 2018. So these the colored area are showing the current distribution of this glassy-winged sharpshooter. I believe recently it has been found in around the Kern County. So of course, our wine grape growers in north coastal areas have been concerned about it because this vector seems to be the most efficient vector of the disease in terms of the distribution in the vineyard. So there are other sap-sucking vectors, which is one of them is the blue-green sharpshooter that has been identified as the one of the vector in North Coast area, as well as a green sharpshooter and also a red-headed sharpshooter. So I'm not an entomologist. I'm not going to get into the more onto the vector ideas. I just wanted to give you a broader perspective of the how this pathogen could be moved from one location to another location. Here's another the point for this vector is that this vector has been not as effective as the glassy-winged sharpshooter, but this vector has a wide host range in the riparian areas. So which means, especially in north coastal area where we have a lot of riparian areas, these vectors could go back and forth in the riparian areas. It can even find and feed on the some not only perennial but also some annual plants in the native vegetation. Because of that, that makes it very difficult to have an efficient and sustainable control of this vector. That's why probably we have been having these Pierce's disease on all over the California. In here, I just would like to, I just wanted to show you some of the these common symptoms. Monica also showed you the some of those symptoms comparing with the Pierce's disease in her talk. So as you can see that this is the leaf scorch, which is the symptom on the Chardonnay, which is white variety. The symptoms on the red varieties are quite a little bit different. I'm going to show you those pictures as well. As you can see that these symptoms could be confused sometime with the ESCA pathogen, which is the group of the number of the disease, fungal disease that my lab has been also working on it. So the reason that we have these kind of symptoms is that when you look at the where the pathogen colonize and then cause problem is that in the xylem, which is in the wood tissue. So what they do is that they colonize that physically or secondary metabolite. They cause kind of like the drought effect on the plants. Plants are showing in here like the kind of like the water deficiency, lack of the water kind of symptoms. Some of the symptoms could start between the vein, and most of the symptom could be the leaf scorch, which is also the one of the common name of this disease, leaf scorching disease. It also cause problem on almond, on other the agricultural crop as well. The one on the right is showing the severe symptom of the Pierce's disease on Chardonnay. Here's how the disease looked like in very heavily infected vineyard in Hillsborough area in northern grape growing areas. So when it comes to disease, if it is called, it can cause the serious damage. You can also see riparian areas right around this vineyard that you can imagine how efficient these vectors could be on these diseases. So here's another the severe symptom of the disease. When it comes to red varieties in here, Cabernet Franc, the similar symptoms, but when you see the margin of the the leaf scorch has a little bit reddish. So that's like the very characteristic symptom in here. As a plant pathologist, we call these beautiful symptoms, but we don't say out loud. These are like the showing like the quite interesting symptoms. Anyway, I just wanted to give you an idea how these symptoms are different between the white and red varieties. So at the end, these shoots could be completely leafless. That all the leaf could be dropped off, and then that can cause the damage. Another common symptom is the matchstick. As you can see, the petiole is still standing on the lignified root, but there is no leaf attached to it. As you remember that when fall come, which is the right now, all of the deciduous plants, including grapevine, lose their leaves based on where they are attached to the lignified food, because of the plant is getting into the dormancy, where plant is producing kind of like the suberin to detach those leaves. When it comes to matchstick symptoms, the stick is still standing there, and then this symptom could be before the harvest, before the fall, around the harvest time. It depends on the severity of the disease that you can see. These called the matchstick kind of the symptom. Another common symptom is the green island on lignified shoots on the grapevine. So these are the very common symptoms that you can see. Here's the point. So you don't have to have all of these symptoms once in order to confirm or say that this is the Pierce's disease. It depends on the condition, colonization of the pathogen, and location. Each one of those or all of those could show as a symptom on Pierce's disease. Sometimes Pierce's disease may be inoculated or colonized in the plant tissue, but they may not show the symptoms because they may not have enough the density within the tissue to show the symptoms. It may be symptomless. It may also show the symptom for next year. Or these symptomatic plants may be infected by the disease for a year before or 2 years before. It depends on all conditions. So you cannot judge based on the symptoms when the first infestation started on these vines. But these are the very characteristic common symptoms of the disease. Shriveled berry is another symptom. In here you can see the shriveled berry symptoms on both white and also red varieties. It's a quite devastative when you have the heavy infestation on the grapevine. So that's the damage that you could see. So I just want to address the elephant in the room, olive decline in Europe. That has been all over the news for several years, and then another Xylella fastidiosa. But this time, Xylella fastidiosa pauca, which is the different subspecies that has been found, causing the olive decline in all over Europe. It started in Italy, and there are a lot of publication has been come out that they identified with this disease. It's also associated with other factors, secondary or environmental condition as well. So we don't have this specific the subspecies in California or United States, I would say. So they also have different group of the vector. Their vector seems to be more efficient than the vectors that we have in California. So that could probably making this disease more severe on olives in Europe. So this is the number that I found online. Again, Pierce's disease is one of the important problem that could cause millions of dollars. Now we have a Pierce's disease, the task force in California to get work on that. So how did I get involved with this project? So as a mycologist working on the fungal diseases, as an extension specialist, of course, my job is to identify the possible the control methods for our growers, especially grape growers in California. So this is Pierce's disease board funded project. This is the, as I mentioned you, the first years of the projects. So develop or find out the efficacy of the biopesticide, available biopesticide to control or prevent Pierce's disease in California. So when we look at the study design, in this study, what we did is that we used one of the bacteriophage, which is a viral, the strain that could attack or colonize or kill bacteria. So these are all known scientific phenomenon. One of the company has identified or came up with a product. Actually, that product is right now registered and also on grapevine. But in this trial, we wanted to try to do the field trial and then compare with the other possible biocontrol agents. In this trial, we try the product is called the Xylaphi PD. We tried their label rate to compare to get you some more information. We also try it in comparison with the other biocontrol agents that we try in this experiment. So other biocontrol agents that we use, which is one of them is the Parabucholderia phytophyrmans, which is a very well known a group of the bacteria which is gram-negative, and then that is known as colonizing the xylem vessels or the wood tissues of the plant and protecting or causing beneficial anything to controlling the diseases of the woods. This particular strain has been found in root of the onion. And our colleagues, Dr. Steve Lindo from UC Berkeley, has studied on this bacteria and has done some work on application of these bacteria with the injection, with the spray, and also application in the greenhouse. He has gotten some positive, good prevention and controlling of the Pierce's disease. So in this trial, we also wanted to try these bacteria as well. So other two bacteria, Pseudomonas viridifera and Parabucholderia, Achromobacter xylosadians and Pseudomonas viridifera, these two bacteria has been identified by another good friends and our colleagues, Dr. Phil Rosausen laboratory, who has been working on this, looking for the beneficial endophytic microbiomes of the grapevine. And he identified, or his lab group himself, and identified the two of these bacteria that has been working, preventing and controlling the Pierce's disease. And he has published a number of the publication. So in this trial, we also wanted to include that. Not only we applied these individual bacteria or our bacteriophage, we also wanted to combine these individuals with the phage to see if they have any combined effect. One of the reasons that we wanted to combine these is that when we look at the mode of action of those biocontrol agents, especially these beneficial bacteria, so they tend to colonize the plant tissue and then increase the defense mechanism of the plant tissue against the Xylella fastidiosa. That's like the primary mode of action of that. So we just wanted to have that preventative action of that. And then xylophy, which is the bacteriophage, are known to colonize or attack the bacteria, to kill the bacterial tissues as a disease of the bacteria. So we also wanted to see the curative function of it, and then we wanted to compare individual and also combined effect of it. Of course, in our trial, we also wanted the positive control. We artificially inoculated these the vines with the Xylella fastidiosa, one of the active strain, and then we wanted to challenge them to see, and then as well as we had the negative control biopesticide. So this trial has been set up in our field in UC Davis Armstrong Vineyard. We used Cabernet Franc cultivar, which is 11-12 years old grapevine, which shows a very good symptom of the Pierce's disease on artificial infection. We used like one of the recent active strain of the Xylella fastidiosa in this trial, and then we used the randomized block drawing, and then we used 10 wines for treatment, and then we randomly choose 4 shoots for each treatment, and then we apply these treatments on those shoots. Again, in this trial, we only focus on the injection of these products, and so there are other methods that needs to be exposed. We are planning to expose other alternatives in our next year trial. So this is the simple our trial design that we showing that the randomized block design. So what we did in this experiment is that we injected these the biocontrol agents in the shoots, base of the shoots, from between the first and second node, as you can see here, and then later on we applied the biocontrol agents and then come back with the injection of the pathogen. So for the injection, we used this kind of like the injection the device that was developed by the Xylophy company, and it's quite efficient when it comes to the injecting the small diameter of the shoots. Again, this is the experimental design. We applied these on the individual shoots. We haven't come up with the design to how to most effectively apply these biocontrol agents in the plant yet, just experimental. So this is the one that shows the delivery method. This is the short movie that I made it to show you how this device could distribute the spray. So it has a needle opening in the both side. When it goes into the xylem, it just spread the either bacteriophage or also biocontrol agents. We use the same device, as you can see. Molly is showing how we did the injection. So in this, again, we use the green shoots just for the sake of the experiment. This question may come in your mind that at the end, if everything goes well, are we going to inject every single green shoots? No. So that also needs to be the address that we are going to be coming up, that kind of things that could be injected either on the cordon or spurs, or even we are going to try the trunk of it. So anyway, so those are the something that needs to be studied in the future. Here's the one of the one that shows where we puncture both biocontrol agents, also inoculated the points the plant recovered or produced the callousing to recover from the mechanical injury that we made for injection. So And then what we did is that, as I mentioned, that we applied, challenged these spores with Xylella fastidiosa by drop puncture, which is a very common method. What you do is that you put the bacterium solution as a drop, like as you can see. You just make a needle wound in the xylem. The capillarity effect of the plant just sucks into the bacterium inside, and then from there bacteria can do its own tissue and then colonize it and then cause problem. So here's the very simple study timeline that shows we applied the biocontrol agents starting in first week of May, and then sorry about that. And then following day we came back and then inoculate with the Xylella fastidiosa. And then in some of the products we also applied the second application and then moved on. And we first evaluated the trial in first week of August, and then we did multiple evaluation based on the some of the literature that has been that guided us. So this is the sample that one of the positive sample that shows us that how this disease could show symptom actually. So when we first start seeing these results, we were not planning to present any of these the results, and then we had a field day because these results were like the one that was showing like the efficacy of these biocontrol agents and all the phage. We just wanted to share this information. Anyway, we had a field day to show this information. Some of you may or may not attend it, so we are going to be repeating same thing. Anyway, so what we did is that we did the disease rating starting at the first week of August. So later on, we also adapted a protocol by Steve Lindo. By counting the number of the leaf in each shoots, and then also each symptomatic leaves were counted. So that was very efficient results. And then Molly has done with my lab crew all of those works. After 17 weeks, we also collected leaf petiole samples to confirm presence of or not presence of the Xylella fastidiosa by qPCR. Here are the preliminary results of the 1 year of the results that shows like the one on the left, percentage of the Pierce's disease on the disease positive, which is quite about 45% of the disease that you can see. The second one is that the xylaphy, which is the bacteriophage, in the label rate, which shows quite significant reduction of the disease. And the one that we also saw that one of the biocontrol agents, sorry about that, biocontrol agents shows also very significant reduction of the disease control, which is very exciting results. So when you look at the results from here, is that this is the combination of the one of the biocontrol and then xylaphy, which is the one of the best looking the results in terms of the disease reduction. As I was telling you earlier, that when you look at the mode of action of these, both think that it's kind of like makes sense to get these kind of results. Anyway, some of those individual other biocontrol agents also show a significant reduction. Some of them didn't show, and then which shows to like the more disease severity than that one. Anyway, so this is the healthy control that we didn't inoculated anything on those, and then this vineyard is as we know a Pierce's disease-free vineyard that we have been working on it. So when we compare with the quantification of the qPCR test for the Xylella fastidiosa, it's quite matching with the results that we recover from our first year results. That's interesting that we also identified the very low rate of the healthy control, as much as we know that these vines are not inoculated, but that's also that kind of things that shows. Anyway, so these are again, I want to stress out, these are the preliminary results. This is the 1 year of the results of our trial. We are going to be repeating these trials 2 more years. Plus, we are going to be doing the similar trial on heavily infested commercial vineyard in Sonoma area as well, to see if we apply some of these in a larger scale, to see if we can have the control in real life. So conclusion of the trial is that all of the biopesticide used in this study showed the significant either prevention or control of the diseases. And the vines treated with the xylophy, which is the bacteriophage, also showed the 50% of the fever foliar symptom, as well as the presence of the Xylella fastidiosa. And when we combine these biopesticides, they perform much better than individuals. With that, I would like to thank our collaborators, AP Infotech, and also I would like to thank the CDFA Pierce's Disease Glass Wing Sharpshooter Board for funding this project, as well as UC Davis and Plant Pathogens Department, who let us use their grapevine or vineyard for this experiment. Not about this one, but I also work on the other the common grapevine diseases, including powdery mildew, botrytis, sour rot, grapevine trunk diseases. We have a lot of field trial that we have been doing that since we took over this position from Dr. Gubler's position. So if you are pest control advisor, vineyard manager, and if you are about to set up a program for not only Pierce's disease, not only powdery mildew, botrytis, and grapevine trunk diseases, please visit this lab website. You will see all the recent studies about the reports that will give you an idea what kind of program or what kind of new products, including some biocontrol agents, as well as the organics, are present to help you developing your own program. So with that, I would like to have questions if you have any.
Okay, Michelle, it will be coming on to ask the questions.
Yes, hello. The first question is, how can you definitively tell the difference between Esca leaf scorch and simple heat leaf scorch in clay soils?
Very good question. That was my point, because both of the diseases are colonizing the xylem vessels, plugging those xylem vessels, causing kind of like the drought symptoms. But common Esca symptoms are also known as tiger stripe symptoms, which is the one that shows like the between the veins. I would also say that the Pierce's disease show more scorching around the leaves if you have to. But again, there is no definitive description. They both show the similar symptom. But Esca disease don't show white island, the matchstick, and others. Okay, Esca also on the fruit shows measles, because Esca group of the pathogen produce secondary metabolite. Those metabolites cause measles, and then also different symptoms, but not the Pierce's disease. Okay.
Okay, so the next question is, phages are extremely specific. Is the phage product a single phage strain or a mix of strains?
Wow, you got me. Okay, that's not the question for me. I believe as much as I know as the single, but I might be wrong. That question, please send me an email. I am going to address that question for you. Okay.
The next question, does the injector have to be cleaned between injection?
Yes. So we haven't gotten that point yet. Yes. Because you are putting that injectant in the xylem of the plant, and that xylem injector could be contaminated by not only bacterium, but also there are a lot of viral diseases, including some of the ESCA that we are talking about. That could be the potential injection. Yes, at that you have to clean those injectors. Again, this is the research study. We are not recommending anything at this point. We are just sharing the information with you guys. This is not like the, hey, take this and then go move. That we are not there yet. So this is the first year results that I am sharing with you. Anyway, yes, at the end, we are going to come up a kind of protocol that how you can spray. Basically, you can use either ethanol. For the ethanol may not control the viral diseases, but again, hasn't been confirmed yet. But we will come up with that the point. Okay, it could be either bleach, certain percentage could be clean. Yes. Bottom line is, yes, you have to clean from at least from wine to wine.
The next question, will there be any work done by foliar trials or biologicals for PD?
Yes, I would love to do that. And then I know that Steve Lindo has done studies, has found some positive effect of it, and then he also explained how those biocontrol agents may enter into the plant, either stroma or natural opening in the plants. I would like to do that as well. Yes, next year. As well as we are also working on how we can apply these in the soil. So these biocontrol agents are produced in the laboratory, and then we are thinking of that, how we can ferment these biocontrol agents in a larger scale. What if we give them as a soil drench? What's going to happen? Are they going to be picked up? Kind of things. Think about that. One of them is found that one. So we are going to explore that part of it as well.
Have you had any success with Rime on trunk disease?
Okay, it's not related question, but you gotta watch my another talk that I am giving it. Yes, for the Rime, just to give you that brief information about it. So when we apply Rime is the fluorophol, the fungicide that has been used for many other diseases, including grapevine trunk diseases. In our field trial that we have been trying it, we have found out that when you apply them direct onto the pruning wound, they have a good efficacy of protecting the pruning wounds. We have also explored of injecting, forget about what I said, drenching in the soil as a soil drench that could be possible to pick up, and also applying on the pruning wounds. We have get some positive results. Some of those results I'm gonna put in that presentation in my lab website, and then you can have an idea. Yes, there are some efficacy on grapevine trunk diseases, as well as we have other biocontrol agents and also other fungicide that has been that experimentally working on that one. So yeah, go to my web, the trunk disease talks or presentation, and I just gave another talk yesterday in here in Sunday. So yeah, get that information or send me email. I'm gonna send it to you. Okay.
Once the bacteriophage is injected, does it induce cascade signaling in the grapevine?
Okay, so that information is beyond my knowledge. What I know is that the phage doesn't stay in the grapevine for a longer term. That requires application what is once a year. That's my understanding from the products. And again, send me email. I'm gonna get that information to you as well. You are asking me the technical information about the registered product, so I don't want to get into the thing. So okay, well, what else?
How durable is the effect of the phage application protecting the vine from XF?
Again, in this trial, we started evaluating our trial beginning of the August, which is the time that plants start showing symptoms until before the harvest. So all the data that I show you during the whole summer, that means in that whole summer, whatever it is, that was able to work. But keep in your mind that we also inoculated the pathogen once. In a real life, vector may go back and forth. We have no idea. So we are going to be doing that one in a commercial setting as well, so we don't have it. But anyway, my understanding from the company's product is that so this is not something that you can apply once and then forget about it. So you have to apply periodically. That's why the one of the goals that we also wanted to include in this trial, the biocontrol agents, that they have a better chance to stay in the xylem tissue longer than we expected, because originally two of the bacteria were isolated from the xylem tissue of the grapevine, which is the endophyte that we expect, and also the one that was isolated from the onion also showed that the colonization of the xylem. So that's why we are comparing these the product together, which one is going to work better. So what we are going to do is that the next year we are going to keep this trial as if going, and we are going to set the second trial for the next year in the different block. So that way we can also do the longer observation, second year observation in the trial that we set up this year. Okay, so more. So what you need to do is that come to my field day next year.
There's one more question. Any data trialing lime sulfur as a post-pruning ESCA treatment?
Very good question. Thank you for asking that question to me. Okay, so late Dr. Gubler has done some dormant lime sulfur application. I want to emphasize that the dormant lime sulfur, you cannot apply the lime sulfur when the plant is green. You can burn it down your plant. That's one. Second one is that unfortunately I couldn't find any data that dormant lime sulfur application could target fruiting bodies of the ESCA or grapevine trunk diseases. Because of that, I don't have any data. I haven't done any research. I have heard Dr. Gubler has done it. That's why this question probably coming out. I do not recommend at this point, based on my experience and also based on the experiment that I haven't done. So what I can do that this is like the second or third question that I have been asked. What I need to do is that I need to do experiment on this. So I might do another trial this winter to target some of those fruiting bodies to see if there is an efficacy of this. At this point, no data.
So people are wondering, how do we find out the date of your field day?
Outdate. Oh, follow me on the Twitter. So that's one. The second one is that all the information in my, okay, here's the homework. If you are pest control advisor and vineyard manager, if you are doing the vineyard thing, you gotta memorize my lab website, okay? Eskalen lab website at UCANR. And then we put all our information, including trial data, the report, everything is that will give you an idea. We post all our information. I post all of my field trial, especially I'm not active on the other social media, but the Twitter. So I put all my talks information, where I'm going to give talks, what I'm going to talk about it, all the field days right there. So if you are there, if you are not the Twitter person, just look at in my website. Here are the rule of thumb. Are most of the field trials for powdery mildew at the end of the July. For botrytis, we haven't done it, but next year we may do it at the end of the July, beginning of the August. Again, at the end of the July, before the harvest, just keep an eye on me. That I'm gonna let you know about field trials, and then come. We may provide free lunch.
Okay, so there are no more questions. So thank you very much.
Thank you for having
Thank
me.
you, Akif. Do you mind, do you have your Q&A box open? If you could type in the address of your page?
Oh
Is
yeah,
it
I will do that.
Eskalen Lab?
Yes.
If you're able to type it in, then
I will do it
attendees
right
would
now.
be able to easily go to your page.
Yes, I will. And then if you Google my name, I'm probably the only Akif in California. You will find it. I'm gonna put that name right there. Okay. Thank you.
Okay, great. Thank you so much, Akif.